Okay, thank you, Annick, for inviting me. I'm very pleased to present here. Is that one better? I have a bit of a silent voice. I'll try to speak up. Like DJ said, my name is Lucas Evers. I work for Waag Society, Institute for Art, Science and Technology in Amsterdam. And I got appointed there about 7 years ago, and one of my first things was to work with Adam Zaretsky, a bio-artist. Some of you know him. But maybe it's interesting first to say something how I got myself, to this. I'm educated as an artist. I studied politics for a number of years. I didn't get a degree in that. And I worked at a debate center in Amsterdam called the Bali, Center for Culture and Politics, where due to some conversations with Guy Bunting, I was interested in what was happening in biotech. And at that time we had a number of ministries that had the need to organize a societal debate about biotechnology in society. Actually, what they wanted was a yes from the people to introduce GMOs into the ecology and take biotech research forward for agricultural production. What they did was consult women's groups that were more interested in exchanging knitting patterns than being busy with real technological questions, but they brought the whole story like, don't worry about the risks. We have everything in hand, and we're going to enhance agriculture so we can feed the world better. So a very positivist story, and I tried to set up some counter debate because I wasn't really happy with their approach, and I worked with Greenpeace in the Netherlands. They took off with their Frankenstein food campaign, in which they were showing pictures of salad leaves that were, according to them, enhanced with a gene from crab that make the salad more crispy, with the line underneath it, do you want to eat Frankenstein food? That hit the jackpot for them because the whole bias of the Dutch government was biased to the other side, and the whole public said no to GMOs, like the rest of Europe did. And what happened was that a lot of companies that were doing biotech research in the Netherlands left. So what the government wanted to do caused the opposite, by not wanting to have a real societal debate. Yeah, that's how I got interested in that, and at that time I wasn't looking at art that much. I saw Eduardo Kach's work, Alba, I saw George Gessert's work, but I didn't have my own framework yet for that. So and there was no framework within Dutch society and funding schemes for cultural institutions. So that didn't take off then. I organized some debate, but not with the impact I would have loved it to have. Then years later I came to Waag Society, where we worked with, well, it's an organization where we have this tagline that says we use creative technology and technological creativity for societal change, which is quite ambitious, I think, but it summarizes a bit what we do. We operate in digital technology a lot, and biotechnology is a new field, or was a new field for us at the time. So I developed quite some projects with bio-artists, do-it-yourself biology, biodesign, etc. I'm also in a project where this plays a role together with a DJ called Knowledge Incubation for Innovation and Creativity in Science, KICS. And I closely work together with Anna Dimitriou. What I'm going to say something about is what we do with the Open Wetlab, but also show a few formats of the way we work and how that causes let's say productive misunderstandings about what science is, what art is, what do-it-yourself is. If you ask me, the most positive situation is that you have those mixed attitudes that are very critical of what art is and what science is and what do-it-yourself is and the whole politics around it, so that we can approach certain topics and questions in society in a different way. Now, the Open Wetlab. You see my colleague here, Pieter van Bohemen, who is, I think, mixing a gel in which we can run DNA. The Open Wetlab is a very small space where we have some equipment like PCR machines, centrifuges, microscopes, and some Turing equipment, in which we do workshops. We have artists working there. But I always also say that we are not only that space, but also a little network of people that have connections with a lot of creatives, with artists, with policymakers, and also with bigger research labs at universities where the real stuff, the real synthetic biology happens, etc. One of our formats is it used to be called the Designers and Arts for Genomics Award. Now it's called the Bio Art and Design Award because that is easier to say. It's a better title. I started that with the Netherlands Genomics Initiative. That, by the way, was the institution that could spend those millions that were reserved to be spent on biotechnological development after that so-called societal debate that I was criticizing so much. And what the Bio Art and Design Award is that people can send in, artists, designers can send in a brief idea that then is put forward to scientific communities, and if they think it's nice, we have a matchmaking round. They start to collaborate, and the best ones of those are judged by a jury made up of curators, artists, but also scientists, and are awarded 25,000 euros to be developed over a period of 6 months. Yeah, we had one example in there. It was I think the title is always difficult to pronounce because it says something about the velocity and the impact strength of a bullet of a gun. It's about it's a work by Jalila Saidi. She proposed to make bulletproof or bullet-resistant skin. Because she had heard they were producing they were producing a protein in a transgenic goat at the University of Utah, I believe, to make spider silk to produce an alternative for Kevlar, for bulletproof vests. And she thought, well, why can't we modify a human? That was an artistic question. That it produces, instead of hair, this silk that makes the human itself bulletproof. A quite speculative question because we are not allowed to work with humans in that way. But what was interesting is that she went really doing it, and they made in vitro human skin with a patch of silk in it, brought it to a shooting range and shot at it. and it withheld the bullet. Now that was not innovative because we still don't have a human then, but it was interesting that the dermatology department of the academic hospital of Leiden found that it was interesting for further research in order to have a new matrix on which the human skin patch could grow for transplantation, because the skin dissolves the silk after being transplanted. To say something about the Bio Art and Design Award, there's a lot of interesting projects in there, but it's also sometimes problematic because it is, you find a lot of kind of science communications in there that, if you ask me, appropriates arts and artistic production for that purpose, which doesn't lead to art that I think is very interesting. You already feel that I want to criticize this project a bit. Ergo Sum of Charlotte Jarvis. What she did was propose to use IPS. It's not IPS, it's the induced pluripotent stem cells that just got rewarded the Nobel Prize, and she proposed to have some skin of her taken, some bladder cells and some blood cells, and you have those made into beating heart cells, brain cells and blood vessel cells. And Aniek was already saying something about this project, In Potentia, of Guy Benari, that also has a nickname. It's called Dick Head, which I think is very humorous. Yeah. And because it's humorous, you can say a lot about the very boring aesthetics it has, if you ask me. Because it's humorous, it's much more, it has a very different relation to the science it is working with. It's critical of the science, it's questioning the science, and the work of Charlotte Jarvis is not so much. She has made a lot of other work that is much more critical, much more in-depth, that I appreciate much more, and that was the work with the apples and the Declaration of Human Rights. That was inscribed in the DNA of a mold that was living in the apple. Well, science communication, it's interesting that it's all projects that are having a critical or sometimes problematical relation to techno-scientific development. This is another winning project of the Bio Art and Design Award by Zach Denveld and Kat Kramer of the Center for Genomic Astronomy. And they worked with the Center for Healthy Aging, making scenarios of our genomic food futures. And this is actually a sort of critique on the fact that producing insects to feed the future world population is regarded a novelty and might save the world. They researched Dutch literature history and found a book about a little boy called Bartje who didn't want to eat beans, and they found statistics that bean consumption in Europe went down when wealth had risen, because beans were not regarded as something that was very tasty. So they are propagating to eat more beans instead of eating more insects, and the protein found in there is making you live longer. This is also interesting because they found that Starbucks is producing a coffee cup that is bigger than the stomach of the average human. So and here again, there's also a lot of humor in there. If you listen to Zach, I would he's a very good presenter if you ask me. This is another result of the Bioart and Design Award. Interestingly enough, they didn't bring it to a real object that could be exhibited, and of course, if you work with a science communications partner, they love objects that you can put into an exhibition. This is interesting. They didn't make it. They want to make a mirror that was 3D, showing your portrait once you were photographed by a liquid that had bacteria that were magnetotide, that were magnetic, floating in that medium. That they would give it a relief by magnetism, so that it had the relief of your portrait. The technology was so complex that they didn't make it into a real prototype for the exhibition, but what is very interesting to know is that they are still working with the scientific group of AMOF that works with molecular biology, and they're still working on far beyond the project. What I wanted, I'm mentioning this because for me this is a kind of very interesting real collaboration where they are where the process itself is also very important, and not only the project, the object that comes out of it. We also had a project called StudioLab that is finished now, so don't worry about the title. In which we worked intensively with Anna on the Trust Women Artists project, which we hope to continue soon. And we worked intensively with Adam Zaretsky and Rich Bell. Rich Bell I come to later. What is interesting about this project, Adam has been working with a large research consortium called Biosolar Cells that researches how we can enhance photosynthesis in order to produce more biomass in a more economical way. But also look at synthetic photosynthesis. He has been researching how vertebrates like zebrafish are injected with an agrobacterium that makes them partially photosynthetic, but also looking at that there is a mortality rate of 95% in that experimentation. And what we did that experimentation at a big music festival where people could micro-inject zebrafish embryos and were explained all the ethical complications around that. A lot of the people simply did it and were not very scrupulous about the fact that they were actually killing an organism at the beginning of its life. That is interesting because we try to do debates as much as hands-on as possible, because we have the feeling that people behave in a different way within a debate setting like that. And this micro-injection, where we had a lot of discussions with the people that were doing it actually, was a very fine example of that. This herbarium of Adam Zaretsky also contained a mustard seed plant, or it was supposed to contain a mustard seed plant that was genetically modified, and it was taken up into in an exhibition called Yes, Naturally, at the Municipal Museum in The Hague, in the Netherlands, and we prepared a permission a request for the Dutch government to exhibit GMOs for art's sake in a public venue. And here you see one of the result, because if they approve, they have to publicize that in the state newspaper, and then people that object can give a reaction, and then that whole objection process starts. It took very, very, very long before we got an answer of the state, because they didn't know what to do with it. And I was present at a meeting of the advisory board for genetically modified organisms to the Dutch government, COGEM, and I found out that 3 ministers, actually 3 secretaries of state, that is the second hand of the minister, of 3 different departments for infrastructure and ecology, for education and science, and for economic affairs. They had sat together 2 times to discuss this artwork and what they had to do with it. It's interesting that you're asking for permission that is based on certain rules and regulations, and you're exactly ticking the boxes, because we worked with a biosafety officer that was saying, okay, we use triple containment, we use this and this and this and this, then it's perfectly safe. Then there is no technical reason why they should not approve. But they had so much political doubt whether they should or should not approve, that they waited with approval until just the end of the exhibition. And after the exhibition, they said, well, we approve. But it was too late. So we're going to re-exhibit the work and then ask anew for permission and hope to have a reaction. Interesting, at the COGEM there were people that have a farming background. They come from the Agricultural University of Wageningen. Farmers tend to keep their innovations secret, so they said, we should not get involved with the arts at all. And the other part, that was the people that were professors of ethics. They said, well, if you're saying that, you are involving with the arts. So that debate is still going on. And another project is called Trust Me, I'm an Artist, that we did with Anna Dumitriou. I think there is enough, and with also with art object-oriented, with Adam Zaretsky and with Neil White. So I'm not going too much into that. Didier, how much time do I have still?
Let's say 5 big minutes.
5 big minutes, okay. This is a work by the Center for Post-Natural History, Rich Pell. He collected so-called post-natural species of the European Union that were GMOs, that were genetically modified mice, but also a diorama of the seed vault in Spitsbergen. And he placed that in an exhibition object in the anatomical theater that is in our building, the Waag building, where they used to do public dissections of hung criminals. So there was an interesting connection with that project there. I think David was already referring to Drew Andy. Drew Andy once said that we shouldn't use the term do-it-yourself biology, but it should be about do-it-together biology. So we organized a series of workshops that had that title, Do-It-Together Biology. And it was basically trying to bring together the general public, scientists, artists, designers, even policymakers, into those situations where they were together touching the technology and debating it at the same time, whether we should go for it or not. Yeah, Instructables published a file about a bioprinter. about a year ago, and that went viral. So I got a request from people in my organization like, hey, they're able to print living materials. Why aren't you doing that yet? So I looked at it, and what BioCurious did was using an agar medium with bioluminescent bacteria, printing their company logo. We started to make their bioprinter. That was difficult because they hadn't left really useful instructions in Instructables, and we had someone else hacking an Ultimaker. We didn't come to much results, but it was very interesting to see that at least in this way, bioprinting within the do-it-yourself community is also something very fashionable that is getting a lot of attention without being much, I would say. Another workshop I thought was very interesting was about making competent cells, and we did that together with Howard Boland and Laura Sinti of StudioLab. And competent cells, who of you knows what a competent cell is? Please raise your hand. Okay. A competent cell, I learned that during that workshop or before because I said, yeah, let's do it. Is a cell that is bashed upon with protocol and chemicals and so forth, so that the cell structure becomes permeable and it can take up foreign DNA. Is that a correct summary? Yeah.
En français, une cellule compétente, une cellule qui a subi un traitement, tu m'arrêtes si je dis des bêtises, qui va donc pouvoir récupérer de l'ADN à l'extérieur. C'est bien ça. Est-ce que... Une cellule compétente, c'est une cellule qui a subi un traitement de façon à ce qu'elle puisse aller récupérer de l'ADN à l'extérieur de ces barrières qui généralement l'en empêchent.
Something that is done in laboratories very much, a lot, and what you see here is a contract they had made for all participants to understand that they were not making a transgenic organism on purpose. We were using E. coli, and if by chance in the liquid they were floating would be foreign DNA, the competent cell would swallow that more or less and become transgenic. So they conceptualized the whole procedure of making competent cells by also bringing in this notion that you are doing something that is close to, that is bordering legal and illegal. It was also very interesting because they had given us manuals and instructions, and they had done it before us, and then they gave us the pipettes to do it, and a lot of people were not acquainted to the pretty precise work, the trained work you have to do with a pipette, so they couldn't do it, and they were scared of that simple piece of equipment that showed them what it means to do lab work, that there's a lot of manual work playing a role in there. And it's also interesting to see that you're making a transgenic cell for an average person is pretty sensational, but you don't see much. It's what Oren Katz calls the aesthetics of disappointment. You do a lot in your lab, but there's no real tangible results, except for some test tubes and little bits of liquid. Here you see a dinner that also has a reference to the disembodied cuisine of Oren Katz, where they had frog steak. This is beef steak proposed by a Dutch researcher. You see him there, the guy in the white shirt. Mark Post, who got a lot of money from Google to research how you make in vitro meat. So that's an artistic concept going to the market, more or less, because I think Symbiotica came up with the first time with it. But anyway, they're making beef out of stem cells of muscle tissue. And recently it was presented in London as a kind of dinner, but not with a group and a control group. They had chefs discussing whether it was fine meat and it was tasty, etc., etc. So it was really mediatized. We organized a day debating what it means to have in vitro meat and how it has to do with our meat consumption and our perception thereof. A lot of the meat we used to eat, like pork legs, you don't find it anymore in a Dutch supermarket. And if you go to have some minced meat, it is presented in blocks, which is very similar to the meat you already saw presented by Mark Post. So we were talking about the other parts of the animal. We were also talking about the other animal. I made a parfait out of mice liver, and here we are preparing rats for consumption. That, by the way, I thought were tasty. Here's the mice. What is also interesting is I wanted to have this workshop because I was told by stem cell researchers that what Mark Post is doing is using stem cells of cow muscle tissue to make beef, and that is a very inefficient way of making beef, because those stem cells rather want to become nerve cells, and you have to do a lot more protocol before they get into muscle cells. If you use mice cells, strangely enough, they do much better. So there was another point of discussion. For me, it was revealing because I didn't know they were researching vegetarian medium to feed the in vitro meat. We made GMO beer out of cattle feed that is on the European market, that is GMO. Nobody knew that. What we are also doing in our lab is having people DNR barcoding the Hortus of the Free University in Amsterdam that's going to close because of budget cuts. Peter, you saw in one of the first pictures, is developing another PCR machine called Amplino that works on a battery, and it's being made for testing blood in the field for people that might have malaria. And we're also trying to make in vitro rhinoceros horn. Few things like that. Why am I telling this? Well, I'm interested to bring together art, science, technology. I'm finishing. In order to learn from each other, but also in a critical way, not often in a way that is done, for instance, by speculative design, with which I have a sort of love-hate relation. Let's see, do I have everything? Yeah, I want to finish with a PCR picture. This is the Open PCR that is made by BioCurious in the United States. But what it does is simply reverse engineer, or simply, I wouldn't be able to do it, but it reverse engineered the PCR machine after the patent ran off. And then they made a new one, which only costs a few $100 instead of the a few $1,000 the lab PCR costs that is standing next to it. We have one like that in our lab. But I think the PCR you see here, project by Paul van Noes, I also think is very, very interesting because it opens the black box of the PCR and tries to understand what it is.
Est-ce que tout le monde sait ce que c'est qu'une PCR?
Okay, yeah, sorry. What a PCR basically does is replicate DNA so that you have a fragment of DNA that you can better research. What the PCR protocol basically does is doing that by adding polymerase to a DNA medium and heating it up and cooling it down, thermocycling. Paul van Noes did this thermocycling on a campfire in the Rocky Mountains, and he got a result. He got multiplied DNA out of it, but simultaneously he was deconstructing the whole history of how PCR got a Nobel Prize and afterwards soon was patented against the will of a lot of scientists that were already using the protocol. Yeah, maybe I should leave it at that. ICT and Art Connect. We're also involved in. I thank you for your attention. And well, what my message is that we need much more critical awareness among scientists and also among artists and do-it-yourself biologists. Thank you.